Okay. Welcome to everybody. Thank you for your presentation. And first of all, I will introduce you to some concept about p38α MAP kinase. p38α is an important, is a protein kinase involved in different intracellular pathways, and in particular, it is involved in the different pro-inflammatory responses. For this reason, it has been proposed as a possible therapeutic target for autoimmune disease and inflammatory processes. Into the cell, we found 4 different isoforms of p38, named alpha, beta, gamma, and delta, with the alpha isoform that is the most expressed and thus the most involved in inflammatory responses. Even though in the last 2 decades numbers of inhibitors have been developed both from pharmaceutical industries and academic groups, some of them entered in the different stages of the clinical trials. Up to date, no FDA-approved drug are present in the market, and mainly due to efficacy and toxicity problems. For this reason, the identification of new scaffolds able to overcome this problem is a field of very interest in the pharmaceutical sciences. Structurally, p38α MAP kinase is constituted by 2 different lobes, the N lobe and the C lobe, connected via hinge region. We found the ATP-binding site between these 2 lobes, and we can classify the ATP-binding site in different regions. However, to the drug discovery processes, the most important regions are those regions not occupied by the ATP, such as hydrophobic region 1, linker region, then allosteric site, because they're less conserved. Well, in fact, given the complexity of the ATP-binding site, we can classify different types of inhibitors. Type 1 inhibitors are those inhibitors able to bind the same region of the ATP, plus hydrophobic region 1. These inhibitors act as competitive inhibitors. Type 2 inhibitors extend their interaction to linker region and to allosteric site. These inhibitors indeed act as allosteric inhibitors. The type 1α inhibitors show an intermediate pharmacophore between type 1 and type 2 inhibitors. In fact, these inhibitors are able to are able to bind the same region occupied by the ATP, by the type 1 inhibitors, plus the linker region. This additional interaction can improve the selectivity and the potency of the type 1α inhibitors. For this reason, this webinar is focalized on this type of inhibitors. The type 1α inhibitors are represented in PGB by 33 co-crystal structures. And starting from this information, the aim of the project were the in-depth analysis of the interactions between type 1 inhibitors and ATP-binding site, identification of the best structure that can be used in the docking experiments, and then use this information in discovery of novel type 1 inhibitors. At the beginning of the project, the 33 crystal structures between p38 and type 1 inhibitors were collected and classified based on the binding mode and the chemotype of the complexed inhibitor. We found 5 different families, and for each crystal structures, we analyze the interactions between the inhibitor and the ATP-binding site, with particular interaction in the group involved in the occupation of the different regions of the ATP-binding site. This analysis led us to identify the group most involved in the occupation of these regions. And we found that the MAPK group is the group most involved in interaction with the linker region, in which perform 2 hydrogen bonds with the glutamic acid 71 and the aspartic acid 168. However, regarding the hydrophobic region 1, we have that the group most involved in this interaction is the aromatic group. In particular, we found that in para position respect to amino moiety, most inhibitors show the presence of amethyl or chlorine group, and this double substitution is generally associated with the potency of the inhibitor. Regarding, indeed, the interaction with the hinge, most of the inhibitors are able to perform an hydrogen bond with the methionine 109. And this interaction was performed with an aromatic nitrogen or a carbonyl group. Indeed, the aromatic ring is the group most placed in the hydrophobic region 1. Obtaining this information from the ligand-based analysis, we looked for the most suitable protein structure. We looked for one protein structure to use that can be used in a docking experiment. In fact, we have 33 different complexes and 33 different conformations of the p38α that can be used in the structure-based drug design on novel type 1 inhibitors. Now, our question was, which one of these, among these structures, is the best performing protein that can, in other words, that can provide the right binding mode of the docked inhibitor. For this reason, we planned cross-docking studies in which, as ligand set, we used the co-crystallized type 1 inhibitors. The molecular docking protocol was built using the BioSolveIT software, and we found very helpful the possibility to integrate this software with the KNIME interface. In particular, this possibility is very helpful to make automatically the process and to allow the data manipulation of the results. The developed workflow was composed by these 6 different steps: the protein preparation and the ligand preparation, that was followed by the docking experiments. All the docked pods were scoring using HIDE, and then filtered to the RMSD calculation. I will spend some other words for each of these steps. Okay. Regarding the protein preparation, the protein preparation were performed using DLIT. In addition, using DLIT, we have also defined the docking parameters, such as binding site definition, that we define as the amino acid with a radius of 6.5 angstrom from the co-crystallized ligands. We consider, if present, the structural water were considered as freely rotable and displaceable. And in addition, we add a constraint, in particular with the methionine, regarding the hydrogen bond with the methionine 109. Well, using this node, after the protein preparation, we proceed to the ligand preparation. The ligand were extracted from the prepared protein and then converted to a SMILE structure. This step was performed to avoid any possible bias due to the experimental pose of the extracted ligand. In fact, externally to KNIME, the SMILE structure were imported in CSAR that reassess the dimensionality of the structure. Well, the prepared ligand were imported into the KNIME workflow and submitted to docking experiments. 10 poses for each ligand were retained and then rescored using HIDE. HIDE is a rescoring function that optimizes the interaction between the inhibitor and the ATP-binding site. in particular in terms of hydrogen bonds and dissolvation energies. After the hydro scoring, the pose can be visualized interactively using CSAR, or as we have done, can be manipulated in KNIME. In fact, we proceed with a selection of the best estimated affinity pose that was submitted to the RMSD calculation. In this node, we perform the RMSD calculation of amount between the experimental pose and the predicted pose in their heavy atom, considering as a threshold to evaluate the goodness of the pose 2.5 angstrom. Well, at the end of the KNIME workflow, all the data were collected in a unique column, and obviously this protocol was repeated for all the 33 co-crystal structures. 27 out of 33 ligands were used because 27 out of 33 ligands were able to perform the methionine, the hydrogen bond with the methionine 139 that was used as constraint.
Yes, it's time for the second poll, and we would like to know if you use data mining tools as well, like Andrea just showed, for example, KNIME or Pipeline Pilot, or do you use other ones? So we would be happy to know which other ones you use, because most of our nodes, most of our software is available as KNIME nodes or as Pipeline Pilot nodes, and would be happy to know if we are missing out any important data mining tool that many of you are using. So please make your choice. It's dropping in pretty slowly at the moment. Only 75% of you have voted. So be so kind to tell us how to improve. And I'm going to close the poll in 5 seconds, so please make your choice. And I'm going to close it now. So as we see, it's pretty balanced between people who do not use any tools or who use KNIME. Some use Pipeline Pilot, and some say other, but did not tell me in the chat box what, so I'd be happy to know this. And I'm going to give back to Andrea for the rest of his talk.
Okay. After the docking experiment, we proceeded to the evaluation of the results. It was performed in different steps. In the first step, we would evaluate the ability of the cross-docking for each protein to reproduce the co-crystallized binding mode, the experimental position of the binding mode of the TIY inhibitors. We use two criteria to evaluate this ability: the RMSD average, that is the average RMSD calculated for each protein considering all the docked ligands, and the docking performance. We consider the docking performance as the number of error, and the number of error was calculated as the sum between the undocked pose and the incorrect, the number of undocked pose and the number of the undocked pose and the number of the incorrect poses. Sorry. As you can see by the graph, the RMSD average is not very different among the different crystal structures. And also it is for almost all the structures lower than 2. However, the number of errors is quite different among the different structures. And for this reason, we use 5 number of errors as an arbitrary threshold to select the best performing proteins, a subset of best performing proteins among the whole set. Well, for this protein, we proceed to the second evaluation step. In fact, we will evaluate also the ability of the protein to have a good agreement among the affinity predicted Ki by height and the experimental EC50. This was calculated using as a parameter, as the height performance parameter, and this analysis led to the identification of one protein that outperforms the others in these terms. 3GFE, in fact, was found able to have the best value in this term, and this means that this protein is able not only to reproduce the crystallographic binding mode of the inhibitors, but also to associate at that pose the right affinity. Well, for this protein, encouraged by these results, for this protein we evaluate also its ability to distinguish between active against inactive compounds. For this reason, we build a second ligand set composed by our type 1 by crystallized inhibitors and a set of decoys, in particular 50 decoys for each ligand that we considered as inactive. This approach led us to the building of our rock curves, for which we calculate the AUC, and we have a good results of 0.91. This means that this protein is also able to top rank the activity, the active compounds respect to the inactive compounds. Well, taken together, these results shows us this protein can be used as the best performing protein among all the 33 crystal structures. And for this reason, this protein, this conformation, were used in the doping experiment. Well, however, during the virtual screening, we could screen a very huge number of compounds. For this reason, we developed a pharmacophore model. The most active compounds for each of the previous identified family was used to build a common feature pharmacophore model in phase. The final pharmacophore model was composed by 4 different features, in particular 2 acceptor group, 1 donor group, and 1 aromatic ring. Okay, at this, from this moment, at this time, we have our pharmacophore model, the best performing model, and the availability of different libraries, such as our in-house library and other commercial libraries. And for this reason, we decided to proceed to the discovery of novel type 1 alpha inhibitors. The first approach that was performed is the virtual screening of commercial libraries. The selected library was the Amminter library optimized for the virtual screening. This library is composed by more than 5 million compounds, and in addition, for each compound, a set of parameters of descriptors were noted. Well, we calculate these descriptors for our type 1 inhibitor set and use these obtained ranges to filter the Amminter library. In addition, compounds predicted as pains were removed, and at the end we have had a final library composed by 3 million of compounds. This library were submitted to virtual screening protocol, in which we perform a pre-screening using our pharmacophore model, and best fitting compounds having a fitness higher than 2 were submitted to the doping experiment using our the best performing protein described above. The compounds that are found by the docking experiment were then filtered using on the base of estimated affinity, ligand efficiency, and to take into account also the efficacy toxicity problem. We add a filter for the off-target activity against PGP and HR. The best 180 from the initial set of 3 million compounds, we ended with 180 compounds that were visually inspected in CSAR, and after this visual inspection was selected, 20 of these compounds were selected for the evaluation of activity against p38α. 10 of them were found active, with the best 4 showed in these slides, and in particular 2 of them showed submicromolar activity. These results validate our vector screening protocol and the synthesis. And for these compounds are now ongoing the evaluation of selectivity profile and of the cellular activity. And when we will have this result, we will plan hit-to-lead optimization.
Yes, then it's time for our last poll. And the question is, how likely are you going to recommend BioSolveIT to a colleague or friend? We saw that we always have, not always, but several times see the same faces in our webinars or the people who try out our tools. And so we would like to know how familiar you are with us and if you had good or bad experiences so far. So half of you have chosen. For the rest, I give you a little bit more time. So please make your choice, and I'm going to close the poll soon. In 3, 2, 1. Okay, now it's closed, and here are the results. That's good to see. We are happy with that. At least not bad experiences so far. So thanks for voting, and enjoy the rest of the talk by Andrea.
Well, okay. In the last part of the project, we focused our attention on the chemical exploitation of an in-house scaffold. In fact, recently we have been involved in the identification of new scaffolds able to act as p38α inhibitors with a good activity and a promising selectivity profile. In addition, the experimental binding mode that could agree with our predicted binding mode showed that this inhibitor bind the ATP-binding site in a type 1 manner, with the fluorophenil moiety placed in the hydrophobic region 1. For this reason, the aim of this last part of the project was the identification, the development of type 1α inhibitors based on our scaffold. To achieve this result, we decided to introduce different moieties able to target the linker region. In particular, we used the amine moiety and the urea moiety that was highlighted important in the ligand-based analysis. In parallel, we would add some other groups, such as methyl chlorine group, to better fill the hydrophobic region 1. A focused library of all possible combination of these substitutions was produced using another KNIME workflow. This workflow is actually a part of the BioSphere workflow provided by BioSolveIT. and can be divided in two different steps. In the first steps, the reagents were imported in the workflow and combined based on the reaction designed in the Marni sketch. For example, in this reaction, we have the virtual reaction that lies to the addition of the urea moiety in the ortho position. The reaction library synthesizer performed the virtual skinning synthesis of all the possible derivatives that were collected in a unique library, and then were screened out the compounds predicted as paints. In this way, we obtained a library of about 5,000 of derivatives that were filtered as first with the pharmacophore model and then submitted to the docking protocol. At the end, the most promising 19 compounds were evaluated for the synthesis with the best 10 selected and that are known and for which the syntheses are now ongoing. Just to make you a few examples, these are two molecules selected for the synthesis. As you can see, the binding mode is very similar to the binding mode of the parent compound, but in addition we have the interaction with the linker region, with both compounds able to interact with the two key amino acids of these regions. Well, in conclusion, using BioSolveIT softwares and in particular the KNIME implementation, we are able to achieve all the goals of this one-year project. In particular, we have developed a KNIME workflow that could hide different drug discovery projects. Well, I would thank all the people that participate to this project and all of you for your attention.
So